We can see it.
Great. Okay. All right. Well, again, and start the timer for me. There we go. I'm Dr. Catherine Zeman, James Madison University. I'm a professor of environmental health science, and I'm the academic unit head for health sciences at James Madison. Dr. Shrestha.
Hello, everyone. This is Junu Shrestha. I'm an assistant professor at Department of Public Health at University of Illinois, Springfield. And I worked with Dr. Catherine Zeman. She was my advisor when I was doing doctoral program at University of Northern Iowa. Okay, so let's start with nitrates in the environment and how nitrate is cycled in the environmental system biogeochemically. So our atmosphere contains 75% of nitrogen gas, and this gas is essential for plants and animals. But these gases, they cannot be utilized in the gaseous form. It needs to be changed into various forms. So nitrogen cycle converts these gases into various forms to be used by plants and animals for their growth. So this atmospheric nitrogen is converted into ammonia by the process called nitrogen fixation. And it is carried out by bacteria that is present in the roots of the plants. And also naturally through the lightning process where this nitrogen gas is converted into nitrates. So these ammonia, when it is converted by these bacteria, they are taken by plants and then it passed through the food chain to the animals. And when these plants and animals die, these nitrogenous compounds are converted into ammonium and then to nitrites, nitrates, and then again back to nitrogen gas by various bacteria and fungi. So these processes are called ammonification, nitrification, denitrification. And among them, like nitrates is very useful form of nitrogen compound in the nitrogen cycle, and it is very essential for the plant's growth.
Person admitting. Okay. Yes, and what we okay. Sorry. Yeah, I had to admit somebody and it's
okay,
kind of stopping the slide. Sorry. So what we want to talk about, how disruption and disjuncture in the natural nitrogen cycle that Dr. Shrestha just described and anthropogenic hijacking of that nitrogen cycle is creating a decoupling of another major biogeochemical cycle, not unlike the major decoupling of the carbon cycle that we're currently experiencing, where we have taken carbon that was tied up in solid deposits deep into the earth, pushed those out into the atmosphere, and created a climate problem for ourselves. In this instance, humans are taking nitrogen that's in the form of nitrogen gas, largely in the environment, and through a process called the Haber process right here, which we began to really implement in the 1940s and on, we've created situations where we have in watersheds that are agriculturally linked, a super amount of reactive nitrogen, a very large amount of reactive nitrogen. So the Haber process takes the gaseous nitrogen and using hydrogen and an iron catalyst at 450 degrees Celsius at 200 atmospheres creates ammonium and liquid ammonia that we can then apply to agricultural areas to increase the amount of soluble nitrogen available for plant growth. Ironically, there was just a publication that showed up in Science a couple of weeks ago looking at the way in which reactive nitrogen is the natural nitrification process. Sorry, it's not going back for me. Every time someone sends in, I have to kind of start the presentation process again. It won't. It's not progressing the slides. try this next. Okay, so what ends up happening is, because of disruption of the carbon cycle, the natural nitrogen fixation process is stalling out. The Haber process and the nitrogen cycle that humans are driving is in excess in watersheds that are agriculturally linked. So we literally have a situation where humans are consuming increasing amounts of reactive nitrogen, particularly in watersheds impacted by agricultural production, while apparently in watersheds associated with natural nitrification processes like Dr. Shrestha described, we actually are starving for reactive nitrogen. So this is some really interesting situations, and it has major implications for the stability of another major biogeochemical cycle and biomes that we depend on, the stability of those biomes and the biodiversity and all of the services that they provide relative to stability of major biogeochemical cycles, the water quality, stability from biodiversity perspective. All of these things are at risk, and we are consuming, particularly in agricultural watersheds, more and more reactive nitrogen in our drinking water. Now that will vary from place to place a bit, depending on how much agricultural activity is going on in a particular watershed. But for human health, that extra reactive nitrogen is a serious concern. On the opposite side, for the stability of biomes, which ultimately feeds into the security of human civilization, that is also a risk. So we have a major problem going on in two of our fundamental biogeochemical cycles, the carbon cycle and the nitrogen cycle. Of course, in the United States, there are regulations to control the amount of reactive nitrogen in the water. Dr. Shrestha.
Okay, so Dr. Zeman talked about the Haber process. So, but we need to know, also we need to know that like nitrates are important in our body, in plants and animal growth. So talking about like natural processes, right? So as I say that it is important, but it is worse if we have a lot of nitrates in our body. So that's the reason why a Safe Drinking Water Act, it establishes Environmental Protection Agency to regulate the concentration of these nitrates and nitrates compound in drinking water. So the maximum contaminant level of nitrates in drinking water is 10 parts per million, and nitrite is 1 parts per million. And it only applies into the municipal water system. So people who are drinking water from the private wells, or they are just people who are drinking water from groundwater, which they have in their private property, they are not regulated, but it serves as a guideline for people who drink private water, as 10 parts per million is the maximum contaminant level of nitrates in drinking water. So although we need nitrate in our body, human body gets nitrate in many sources in nature, such as food items that we ingest. For example, spinach, lettuce, beets, carrots has natural form of nitrates. And in addition, food additives which are treated chemically, for example, meat product are treated with nitrites to increase the shelf life. So that adds a lot of nitrates into our body, which results in various health impact. Most significantly, it impacts in acute toxicity. And in this next slide, I will talk more about Methyl Hemoglobinia, or blue baby syndrome, that is caused due to excess nitrate in drinking water or in food supplies. So when excessive nitrates and nitrites, when it is present in the blood, then the ferrous ion in the hemoglobin, Catherine, can we move? Yeah. So the ferrous ion in the blood, that in the hemoglobin, that is converted into the ferric ion and forms methemoglobin. So methemoglobin, it is not required by our body. I mean, like it just limits the ability of our body to carry oxygen from one tissue to another. So it creates a tissue hypoxia, which is lack of oxygen. So the treatment of methylhemoglobinia is to provide excessive supplemental oxygen to the body and with methylene blue. And it is very common among kids because their gastrointestinal system is not fully developed to metabolize excessive nitrates and nitrate in their body. Catherine, can we go to next slide, please? Thank you. So as I mentioned in my previous slide, that methylhemoglobinia is common among kids and mostly among kids who are less than 5 weeks of age, which creates sudden health impact due to excess nitrate concentration. So nitrates that is consumed by food or by drinking water or even through the medication, it is converted into nitrite by the microflora that is present in our mouth. These, when it is converted, so this nitrite, it forms methemoglobin. And in infants, the main exposure of nitrate is from the infant formula, which is mixed with well water or private water. So excess nitrate, it has like sudden health impact like cyanosis, which is a lack of oxygen, and it can progress to difficulty in breathing and even coma.
Okay, so we're going to really have to pick this up because I know we're at 11 minutes, and I'm sorry, but when people sign in, it somehow is bumping the slides around. Okay, so if you look at the balance of, it's called the volume of distribution, it's the amount of a toxin in your blood versus distributed in your tissues. If you take a look at the natural background levels of nitrate in the environment, without agricultural input, you're going to find 0 to 2 parts per million nitrate nitrogen out there in the water. If you get into areas where there's more agricultural input, it will go much higher, obviously, tens, hundreds even of parts per million nitrate nitrogen. In our body, it's interesting with critical tissue areas, for example, amniotic fluid, cerebral spinal fluid, or plasma, the body works to keep the amount of reactive nitrate, nitrogen, nitric oxide at about 2 parts per million or less. In other areas related to the gastrointestinal tract or the urinary tract, where excess nitrate is being removed from the body, you can see higher concentrations. And there is a direct relationship between the ingestion of nitrate, the formation of nitrite by bacteria in the gut, and the formation of nitric oxide in the blood. So the reactive nitrogen does increase in the plasma when we increasingly ingest it in water. It's a little different situation in food. We probably don't have time to go into that, but if it's a question, we can answer it at the end. So basically, the protective layer, the Safe Drinking Water Act that Dr. Shrestha was talking about, 10 parts per million nitrate nitrogen, is for that infantile methylhemoglobinemia condition, not for long-term health conditions. There are epidemiological studies, not just correlational, but as well as retrospective and prospective cohort well-designed studies with examples showing associations of blood pressure issues, brain tumors, colorectal cancer, insulin-dependent diabetes mellitus, gas immunity issues, non-Hodgkin's lymphoma, ovarian cancer, prostate and thyroid problems. And in fact, when I did a review recently of the chronic disease studies, I found that about 66% of them were finding these relationships at 0 to 10 parts per million nitrate nitrogen. Importantly, that is at the and under the current regulatory level, which is 10 parts per million nitrate nitrogen. That is because that was written for acute infantile methylhemoglobinemia, not chronic disease issues. So we have some major challenges to the nitrogen cycle right now. Plants are starving for it, and humans in agriculturally impacted watersheds are ingesting a great deal of reactive nitrogen. This led to some of our cell culture and toxicology work that you can read about in our full paper that involved taking a look at the way in which this reactive nitrogen and people consuming different levels of it was impacting inflammatory processes in the body by looking at the immune system, precursor for inflammatory molecules. Okay, so we took a look at macrophages here and the kinds of cytokines that they produce. We also took a look at whether or not that reactive nitrogen was having an impact on induction pathways. This is the surface of the cell over here. This is the nucleus of the cell. All of these molecules and connections are the kinds of things that have to be in place just for normal programmed cell death to occur. That's apoptosis. It's cell death without inflammation. All of these bonds are phosphate linked bonds, okay? And this is the nucleus. What we took a look at is whether these bonds were stabilized in lymphocytes that we studied, and also whether there's any DNA break in the nucleus. And we used a technique called Flow's method. I really don't think we have time to go into touch right now because we're running out of time, but Junu, do you want to say something real quick with it?
Catherine, I just your video froze for a few seconds, so I could not hear everything. So yeah, I think we are almost out of the time. So what we got from our research is we can see, so we did we took the blood sample from people and then injected different concentration of nitrite to see whether that was very impactful to the blood cell. And we found that even at lower concentration, there was impact seen in this white blood cell, indicating that there might be some impact, but we have a sample size of 31. So if we had more samples, then we could correlate more about how these nitrate exposure to human blood would impact. So when we looked about cytokine, cytokine number of cytokines, which are the proteins that is formed from our body by pro-inflammatory and anti-inflammatory cells. So those all had an impact. And also the lymphocyte proliferation was also impacted. So there was lower lymphocyte proliferation when we had a higher concentration of nitrite. So we can see some subtle impact due to high nitrate concentration in human blood cell.
Okay, so for example, this slide shows a narrowing of the coefficient of variation, which is a quotient produced by dividing the number and vigor of stimulated to non-stimulated cells in the immune system. These are macrophages or lymphocytes, frontline cells that provide primary frontline immunity and are mobile. This is taking a look at the DNA damage, the percent of DNA damage and dephosphorylation, stimulated to non-stimulated dephosphorylation. And you can see that there's increases at higher rates of nitrate exposure in all the situations. We also saw increases in inflammatory cytokines in each situation. We had a super responder here. We still saw the increase even when we controlled for the super responder. And in every one of these situations, we think that it would be important to take a look at this more closely with larger sample sizes and be aware that these concerns and problems are occurring with nitrogen cycle disruption. And we're out of time now, so we'll stop.